protocol publication, which Amy provided in the invitation, a link for that publication, and Amy can put that in the chat at some point as well. So we have a protocol publication. It was part of a book, and it was one of the chapters, and that gives the additional information. There are also images, which I'm going to show today, but there's also a timeline. So I encourage you to both look at the recording and also read that protocol chapter. All right. So what we have up here on the screen right now is this is a follow-up of questions that we had from last time. We didn't have images, but this will more clearly explain what we meant by the kind of the top down adaxial or the bottom up. So Carrie, did you want? So I'm going to turn it over to Carrie, because Carrie actually does this. I mainly sit at the computer. David, Carrie's in the hood. So Carrie's going to explain in the protocol about this adaxial and abaxial and why the change and when we make the change. Okay, Carrie.
Okay. So when you first cut the cotyledons to do the transformation, and you place them back on the filter paper, they are top-down adaxial. And after the transformation, they're still top-down adaxial. But after they've...
Sorry, I got my pointer. I got the pointer to work.
Oh, okay.
so I can do. So yeah, so as Carrie mentioned, so this is a side view of a cotyledon. So this is the top side of a cotyledon, and then this is the bottom, what we're calling bottom or underneath. So, okay, sorry, go ahead, Carrie.
After the transformation is done though, and it's incubated for the 2 days, when they go onto the media, then you're gonna want them abaxial, bottom down, because as you can see, cotyledons have a natural curve. So you want them placed on the media so that everything is coming into contact with the antibiotics in the media.
And that's especially true. You can see where we have the bottom down. Those are the cut edges that are in contact with the medium. And the reason we do this is because two things is if we, in our pre-culture system that you'll see in the protocol, if we put that top down, if we put it top down, we have more of the surface area in contact with the medium than if we flipped it over. So for that 1 day pre-culture, those little cotyledons would start to dehydrate. So that's why we put the top down first. But then after we do the infection, we flip them, because then you have the cut edges where the agrobacterium got into the cells, so that you have better selection on those cut edges. So I hope that makes more sense than when we discussed this at the event earlier where we talked about the protocol. Okay. All right. So Amy, if you can go to the next slide.
Okay, sorry, just give me 1 second to advance.
Okay, no problem. So this I took right from that protocol chapter that I mentioned. These images are a part of that, along with the timeline. So I thought maybe you guys might be curious that, for one experiment for the tomato line that we mainly use, which is called M82, we use 250 cotyledon sections. Those are transferred to medium every 2 weeks. And what we've estimated is that one person, full-time employee, can do about 52 transformation experiments per year for our M82, and it takes roughly 3 months to recover the very first transgenic lines, and then an additional month to recover the remaining modified lines. So it's kind of like a bell curve, whereas that first 3 months we have some, and then we hit a point where we have a lot. And then after an additional 2 months, it starts to wind down and we're recovering. So from start to finish, meaning from the time we do infection until we've recovered all the plants we want, takes roughly 5 months. And that's with M82 and some of our other tomato genotypes. We've done a lot of tomato genotypes over the years. And so that's basically what we see. Amy, if you could go to the next slide, please. All right, and this just shows, this is something we didn't show last time, but it's just how we're doing this transformation. We have the plate over there on the left, which are agrobacterium colonies after we've done an electroporation and put on selection medium. We're using a liquid culture system. We put in the colonies into this liquid culture. It goes on a shaking incubator overnight. And Amy, if you could click the forward button again. And then after an overnight culture, this is the kind of what we see. This is the agrobacterium growth. We always measure the OD of our agrobacterium solution. And you'll see in our protocol that OD 600 that we're trying to hit. I know some people don't measure their OD every time they do a transformation, but I'm not too sure that for us, that's not what we consider a good practice. So we always read the OD before we do a transformation. And then the next couple of slides are just in case you're curious about kind of how long this all takes. So we start with those germinated seedlings. We have our cotyledons. You can see in these cotyledons, that's the top of the cotyledons down. That's the pre-culture period. And then after we do the transformation, they go to medium, they go bottom down. And then you can see that we have these on, this is actually a kanamycin selection. And you can see that we've got some transformants starting there. After about 4 weeks, what's on the left there image is some non-transgenic controls. We always do controls. And you can see they're not rooting on kanamycin, which is a good thing because they shouldn't. So we've really got a good kanamycin concentration to inhibit that rooting on wild type. And then the plant on the left is a transformed plant that's rooting beautifully on that kanamycin containing medium. And then Amy, if you could go to the next slide. And then we transfer them to soil. This picture here, we used to use these juice containers and cut the tops off, but now we're using water bottles, and they work perfectly well using, we reuse them, so we'll wash them and reuse those. Those little plants that come out of culture, after about 4 weeks, they look like the image on the right, looking beautiful. And then in about 12 weeks, we have this nice ripe red fruit for M82. And then Amy, if you could go to the next slide. So going from an infected cotyledon to getting fruit that we could harvest takes about 6 months. Okay, so that is, just wanted to give you an overview sort of some of the specifics from feedback from last time, and then a bigger overview of the timeline for the process. Right. Okay, so I believe we're ready to take some questions now.
So I have a question. I think I sent 4 questions in during my email, but I'm just going to address one for the moment.
Okay, sure.
So going back, actually, you contributed to a book back in which I published in 2004 on tomato transformation. So in the early publications of tomato, people use feeder layers. I'm curious to know, do you still use feeder layers, or was there a reason why you moved out?
Yeah, good question. So yeah, that was our earlier method where we were maintaining a tobacco cell line called NT1, the NT1 cells, and we were using that as a feeder layer. That was based on a protocol, a publication, even before we started, before I joined BTI. And then at some point we thought, do we really need this? Because we're maintaining this cell suspension, which is extra work. We would maintain the cells in a flask, and we also had the cells as a backup callus in case we infected the cell suspension. So we did experiments where we looked at with and without the feeder layer. And we saw no significant difference between using a feeder layer and not. So that's not something that we introduced. That was based on a much older protocol that was being used when I first came to BTI. But it was just questioning, do we really need it? And we didn't. So we stopped using the feeder layer.
Okay.
Yeah.
Thank you.
Sure.
So another question that was raised actually in the last presentation, people addressed the issue about zeaxanthin riboside, which is a source of cytokinin. And I think anybody who's used it will tell you that it's very expensive. So my question is, there are alternative cytokinins available. If you change over from zeaxanthin riboside, like for example, to BAP, do you see a down in the transformation efficiency?
We have never looked at other growth regulators because again, this protocol was here before I came, and it was based on another protocol, and then they modified it, and it was working so well. I mean, and I know it's expensive, I agree. But like with our MED2, we get 98% 90% transformation efficiency using the zeatin, and yes, it's expensive, but there are other protocols out there in the literature that do use other growth regulators, but we don't have any experience with those. We had a big pipeline for doing transformation, and it just zeatin worked well for us, and we didn't switch.
Okay, thank you.
Sure.
So another question I have is, so I think in your protocol you've listed the use of NPT2 and HPT as your selectable markers. I'm curious to know whether you've used a bar gene as a selectable marker in tomato.
We did. We used it both in tomato and in potato because we were doing potato at one time, and it was really difficult to work with and to get transgenic lines back. And it was a really good effort that we made because we had a group here we were collaborating with that their vector systems had the bar gene for herbicide resistance, and we just we could get material, but it was a lot more work and not very efficient. We really had to ramp up the number of explants we used, and sometimes we had to repeat experiments. So we just asked people to please not use those, use those vector systems with bar. Hygromycin works really well. That's also, I think it's an even tighter selection for tomato than kanamycin. Yeah. So, but yeah, so we've stopped using herbicide selection a long time ago.
Okay. One final question is, so in your rooting media, you don't use an auxin, is that correct? And is that because you have, for rooting, tomato is fairly straightforward?
Yes, it's very straightforward. It roots readily in medium without growth regulators. However, there have been times when we've either done gene overexpression or gene editing where we had some difficulties getting them to root, and we knew they were transformed because we tested that later, and we did add some indole acid to the rooting medium to get those to root. But it's so again, if we somehow have transformants that we're having difficulty rooting and we know they are transgenic, then we'll use IAA. Do you remember the concentration?
I believe it was 1 mg per liter, but I could.
Okay.
Because I think
Yeah.
some stuff though we did do 0.1, so I'd have to look which was which.
Yeah,
I think
but we did add IAA to the medium.
So when you root, do you also have the selectable agents? No.
We do, yep.
You do,
Mhm.
you do keep it.
Yes, we do. We put the same level of kanamycin in with or hygromycin. Sometimes we had to drop, but we changed the hygromycin, but it's in the rooting medium. We have that all the way throughout in the root and into the rooting medium.
Okay, thank you.
Yep.
Tim, I see you
Hi,
have your
Joyce.
hand raised if you'd like to ask a question.
Yeah. Thank you. This is Tim Olmosov from Sequesta. I actually dialed in late and I missed some of the presentation, but I have a few questions. What you mentioned that you always religiously checking the OD of the agrobacterium, and I want to follow up on that from a personal experience that we were doing the same thing in cover cress transformation when we were starting it back in 2016. And one day, the person who was doing it kind of stopped doing it because of some feedback from others. And initially, what kind of pipeline would be affected dramatically, negatively, and it didn't happen. So instead of OD 600, I think, or rather, 0.6, he actually was growing it almost to maturity or maybe to the steady phase, and it didn't affect the transformation rate, at least in cover cress. So I was curious, you didn't mention, you check in the OD, but what's your target OD, and have you looked into different concentrations of the agro?
I mean, the target OD is 0.6. But
Yeah, we certainly looked at target ODs when we started to get the most efficient transformation, and the 0.6 was that sweet spot for tomato.
Yeah. Okay.
Yep.
Well, that's good to know. That's kind of accepted in the field that probably should be the target for everything. But I'm not
Yeah.
sure how much really solid experimental data is there to support that. View.
Yeah, for us, it was definitely true. And it's not just for transformation efficiency, it's also for overgrowth. You know, when I've talked with people and they say, oh, I've got a lot of overgrowth, we've learned that they didn't measure that OD and it was way off. And so they were infecting with a lot more agrobacterium than they needed. And I think for some species, tomato, of the species we work with, and you can correct me if I'm wrong, if we get any overgrowth, it seems to be more with tomato than other systems. And so if people are using an agrobacterium concentration that's way over the 0.6, then not only, you know, it's not just about transformation efficiency, it's about controlling that overgrowth.
That's another good point. Yeah, overgrowth is obviously something that always watch out. And on that topic, there are now new strains, right? Specifically designed oxytrophic strains.
Right.
And we don't have them here, but I'm thinking about bringing them in. And I was wondering what your or other people experiences. There is methionine and there is thymidine oxytroph, and people have different opinions which one is best. Do you have any experience with that?
We do. We're using the Corteva leaf transformation for maize, and we're using the oxytropic LBA 4404. And that works well. We haven't had a problem with overgrowth. I think the selection for that is meropenem. But that's the only experience we have is with that particular one.
Okay, so in your opinion, it's worth using oxytrophic strains?
I guess it could depend on the species. You know, I think it's like we use AGL1 for, you know, for our tomato, and we don't have a problem with overgrowth. But if you use it for maize, you do. So there's really, you know, it's a plant pathogen, right? So it's going to be acting. So we can't say across the board if it's better or worse. And you also have to be, which I'm sure you know, when you're doing oxytrophic strains, is to make sure your efficiency is good. Because it doesn't necessarily mean you're going to get a good transformation efficiency.
Right.
So that's another thing you need to check when you switch.
Right. Well, people who developed the strains claim that the efficiency was not affected.
Yeah, but you know,
it has been
depends.
Yeah.
I'm always, I'm not that trusting.
Yeah. Well, my other question is morphogenic factors also seems like you know, it's not really truly emerging trait. I think it's a trend that does exist. Do they make any sense to be used in tomato or not?
You know, tomato, I love working with tomato because it, you know, there might be, I have a project where we were looking at, we kind of like, what are the genetic factors behind plant regeneration? So I really wanted to do GWAS and look for tomato lines that did not regenerate. We had to screen over 150 before we found 2 that didn't work. So, you know, for tomato, we don't need the morphogenic genes. For other systems like the leaf transformation, again, of maize, they work well. And maybe in other difficult systems, they might be worthwhile. But for the tomato, which we're discussing today, we don't use them. We don't need them.
Well, since you identified those 2 lines, did you follow up on try to understand what's different about those 2 or not?
We are. Yep.
No. Okay.
Yep.
Is it published work?
Not yet. It's in progress. It's in progress. Yeah.
That would be,
Yeah.
that would be
Yeah.
very cool.
It's in the functional characterization. Yeah, we're in that process now.
That's awesome.
Yeah.
And my last question is about the transformation cycle. I think the slide was very useful from infection on the plate to harvesting mature tomato fruit is 6 months. And obviously everyone wants to shorten that cycle. What ideas are being explored to do that?
Well, to me, it's maybe trying in-planter methods and getting away from tissue culture.
Is there any evidence that it worked in tomato?
It will be, there is a paper in Nicosia Benthamiana where they have supplementary by Dan Voitas's group, where I think it's in that paper, or they do overexpression in tomato and they show a method. But it's in the supplementary, but I know there's work in progress that I can't share information, but it looks promising.
Okay. Yeah, that would be cool.
Mhm.
Well, thank you very much for
You're
sharing
welcome.
this
You're
work.
welcome. Sure.
So Joyce and Carrie, there were a couple of questions in the chat.
Okay.
They were related to applying this protocol to other species or other tomato varieties. So I'll just kind of group those together. So the first question was from Billy. How translatable is this protocol to other similar genus like Physalis? And then there was another question from Diego. Are there significant differences in transformation efficiency between the tomato cultivars?
I'll start with the Physalis question. It is translatable for ground cherry species we have found, but it is not translatable for goldenberry Physalis. Because that you would need BA and NAA. But if you're doing ground cherry, yes, it works very well, and you can actually use like less tomatin, but everything else is pretty comparable.
Except the cotyledon versus hypocotyledon.
Oh yeah. The biggest thing is instead of cotyledons for the ground cherry varieties, you do have to transform the hypocotyls, and they're a little more delicate, so you just have to be a little more careful and not damaging them in the transformation process.
And as far as other tomato lines, other tomato types, you know, our gold standard is this M82, which I mentioned we get like a 90% transformation efficiency. And then we have another one that works really well called Elsa Craig that people are using as a model. We do see variation in other transformation of other tomatoes. And what we end up having to do is not tweak the method necessarily, but tweaking the concentration of kanamycin. And to be able to get a good transformation efficiency. That means going lower. We even, for one of our lines, we would take it off kanamycin at a certain point, like when they were little shoots, or no, even when they were at the cotyledon stage. And then go back when we went to rooting, we'd go back to the kanamycin. So that was rare. There were very few that that we had to do that. But in general, like the M82 and the Elsa Craig, if you're looking for good tomato lines that work well, in our experience, those are two that do that have worked for years. We've been using those. I hope that answers the question.
Sorry, what do you, sorry, just a quick question. So where do you get your seeds from? Do you actually produce your own seeds?
Yes, we do our own bulk up of seeds.
Okay.
Yeah.
So are you willing to send seeds out to other people to do research? Is that available? No.
No, it's not that sorry, just gonna cut right to the chase. No, it's not that I don't want to share, I love to share, but it's just time for my group to have to do that, to bulk up seeds. So, you know, we're always happy to, on a small scale, send people some seeds so they could do their own bulk up, but we don't do the bulk up for people just because the reality is that people in my group are funded on particular projects, you know, national NSF or USDA, and we just don't have the time to provide seeds to people.
Okay.
Yep.
Sorry, just another question actually, it is technical. So in terms of the explants, I'm curious to know, so you remove the PTO.
Yes.
I'm just wondering, have you ever done experiments where you've actually have the PTO intact, or at least part of it, and see whether you could improve the efficiency further?
We haven't, and I don't know, as I mentioned this, I had already had this in place before I joined BTI. And I don't know if they looked at that. But you know, it's a good point, Ian, because sometimes that petiole is very regenerable.
Extremely.
Yeah, it is, or where you might even see the regeneration comes from that petiole end. So again, we haven't, but and I don't know if the people who were here that kind of put this in place ever did that. Yeah, but that's a good point.
Dharmendra, I saw you had your hand raised. Do you want to ask your question?
Yeah, hi Joyce.
Hi.
The old question, is it possible to transform the plant with the R genes?
Sorry, Dharmendra, do you think you could speak up just a little bit? It's a
Yeah,
little
thank
difficult
you.
to hear.
Okay, is it possible to transform the plants with the R genes and the different cultivars or the accessions? Means how easy it is to transform tomato. In the past, it was a little difficult. And also, is it possible to regenerate the tomato protoplast, transform protoplast?
Yeah, so about the protoplast, I have experience with protoplast from VFNT cherry that worked really well. I don't know, I haven't used other tomato lines and I haven't looked in the literature to see what people have done. But for my graduate work, I worked with just that one tomato line, VFNT cherry, which was doing protoplast, and it worked well from VFNT cherry. And sorry, what was the other question you had about transforming I'm sorry, I didn't catch it.
So we know sometime it was difficult to transform PTOR.
Oh, PTOR, yes,
Yeah,
that was,
so it
yeah,
was.
right.
Mhm. So is it possible now, or it still is something
Yes,
you
they are, yes, what he's referring to is that here at BTI, there is a group looking at a Rio Grande line, and it was it was called PTOR, and that was a more difficult one to transform in the beginning, but we kept at it and we worked out different parameters and we got it to work. Maybe not as well as M82, but we were able to get it better than what it was initially. Yep.
I'm curious,
Thank you.
the transformation through protoplast, is it the longer process?
Yeah, so for me, I wasn't necessarily, let's see, I don't remember, it's been too long. That I don't know, like from the start to finish, how long that would take and what the comparison is. There's probably something in the literature, but I just don't remember what that process was. I mean,
Overall,
the
just curious. So obviously today you're not using protoplast, but some people I talk to, they love protoplast.
I know, yeah.
Transform everything through protoplast, and I'm wondering what's your opinion on pros and cons of doing that?
Yeah, I think it's this type of thing, the pros and cons to me are the skill level to be able to do protoplasts. You know, it takes some time to do that, having to be set up. It's one thing to be able to isolate protoplast. It's another thing to have healthy protoplasts that go on and divide and make callus. And I know there are certain applications, especially with genome editing, where people want to use protoplasts. And I just find that when trying to train people, it's easier to train them on doing you know, transformation with something like cotyledons and doing protoplasts. Okay, all right, Carrie, Amy, anything else there in the chat?
Yes, there were a couple of questions. I'll start with this one from Marissa. Have you ever seen the transformed cotyledons bleaching after a few weeks?
I have not come across that with tomato.
Okay. And then I actually had a question for myself.
You're
So
not allowed.
I was like, I don't want to abuse my powers as the Zoom host here. But you had that slide earlier with the transferring from you know, the plates into the greenhouse. Are you using distilled water to water those plants out first, or do you just use the tap?
Nope, just regular, just regular tap water. Yep.
And there was a question in the chat about the conditions. I'm not sure, maybe this person could clarify a little bit, but the question was, I believe to get good explant or good transformation, you need to give the tomato good condition, right? Can you share and explain the condition you give to tomatoes, please? So I assume there, again, if you want to clarify a little bit, but I'm assuming this question is about the starting material you use for the cotyledons. And I believe you have the growth conditions in the protocol too, right?
Yep,
Okay.
yep, yeah, it should be in the protocol. I mean, in general, it should be in the protocol, but it's, we keep our culture room at around 24 degrees, and that's, some people are using incubators. When I first came here, they already had a big culture room in place, and that culture room is kept at 24 degrees, but it fluctuates 2 degrees either side when the lights come on or go off. And then we have the lights on at 16 hours lights on, 8 hours off. And then we also have our light intensity is around, depending on where we are in the shelf, it could be within the lights, it could be anywhere from 65 to 75 micro-Einsteins per meter squared, whatever that is. So those are the conditions we use for the cotyledons. I mean, the seedlings to get the cotyledons.
Is the humidity controlled for that room?
No,
Okay.
no, and just in general, I do get asked that a lot, and at least I don't know if anybody else on the call is that, but I don't know of anyone that I know who does tissue culture that they actually control the humidity in the room. Or, I mean, in a chamber, that I'm aware of, so, yeah. There
And then
might be
there was another
some,
question from Billy. Oh, sorry,
Okay.
Joyce, I didn't mean
there
to-
might be some applications, but not that I'm aware of, and we've never done that.
Okay. Yeah, thank you for that. So I'll read this question from Billy. Would you mind commenting on why the protocol uses a pH of 6 in the media when the large majority of tissue culture protocols use a pH of 5.8?
Yeah, some of them in here for. Without knowing right off, most of our medias. Yeah, some of them are 5.8 and some of them are 6, but everything we found 5.8 to 6 for
Yeah.
plant
And
material.
yeah, and again, some of this was in place even before I came. I mean, we didn't tweak the pH when we did this, so I assume that maybe the people before us did look at pH, or maybe they were following something else in the literature, and they used a pH of 6. You're right, and the 5.8 is more common, but that was already part of the protocol at that time.
Thanks. And then there's a question from Elsa asking about what age the plants should be for transformation. So I already germinated my tomato seeds, but they're very small. They're 8 days old. If I use older plants, would that affect the protocol?
I mean, once you have true leaves, that will affect the protocol.
So as Carrie mentioned, what we've seen is once you start to get true leaves from those tomatoes, the cotyledons start to change like physiologically and probably cellular, and they don't respond as well. So even though we give a guideline of 6 to 8 days, the reason we do that is because for the genotypes that we work with, that's before the first true leaves really are emerging from those seedlings. So maybe for your particular genotype, it might be shorter or longer, but it's really important to take those cotyledons before, and that has been something that's been looked at before those first true leaves start to emerge. Okay, welcome.
I just want to add with this, because a lot of people do have issues with seeds, but seed packet to seed packet can be different. And I just want to stress how important the seed extraction method is. Because we have found that people think, oh, seed extraction's easy, and they will have a student or someone do something over the extract for them, and then you just really need to make sure your seed are being extracted from actually ripe fruit, and you're getting your seed clean, because we have found, although fruit can come from the same plant, one packet just has terrible germination and the other has fantastic germination. So, and because you're so reliant on your transformation to have even germination to make everything easier for you, just it is important, the extraction. Don't take that part lightly. And that could also be affect, you know, your seed when you're, I don't know if your seed are not germinating like you expect. It could just be they weren't extracted well.
Yeah, the treatment might have been too harsh.
Yeah,
or-
too much bleach,
Yeah.
not enough,
Mhm.
you know.
Yep.
Sorry, I have a question about that. That's a very good question. I know in some species people use acid during the removal of the pulp around the seed. I was wondering if you could give us some information how to prepare seeds from fruit, please.
So there is that method where people use hydrochloric acid for extraction, but we use, Carrie, if you want to explain.
We use Rapidase. We soak it in I don't remember the percentage right off, for an hour, and then we just do like a 5 to 10 minute 40% bleach soak after that, and that works wonderfully for removing the pulp. I think sometimes with the acids, if the timing's off at all, you're really going to damage the seed more so. So that one is a little, I don't want to say dangerous, but
Detrimental.
yeah, it could be. Whereas the Rapidase, you don't really see that.
So what Carrie mentioned is Rapidase. So Rapidase is an enzyme that's actually used in the food industry, and that helps dissolve the pectin. So we use that for our tomato, our ground cherry, our goldenberry.
You name it.
Yeah,
that's-
we use it for all our fruit.
So if you used acid, what would be the molarity and the time, would you say?
Did we ever even use acid? I don't know if
We
we.
use
In
it
the olden days.
In the
for
beginning,
Soteria,
yeah.
but not. It's
Um,
been a long time. I'd have to go back and look at what was done.
yeah, you kind of put those things out of your
Yeah,
mind.
yeah. I don't like to think about those days.
Hydrochloric acid.
Yeah, I think they do that in the citrus.
Ah,
They
okay.
use acid. Yes.
I see. I see.
That,
And then when I was an undergrad, I have to share this story. I had work study money. My job, I worked with a tomato breeder, and his method for taking off the gel was I would scoop them into these little plastic containers, cover them with cheesecloth, and put those in a very warm room on a Friday, and then come back the next week. And it would just be this fungal mold, disgusting, smelly mix. And then I would rinse off the water, rinse all that mat off, and put them in a clothing dryer for on really low speed. But that was how he used to remove the gel from the tomato seed. So I appreciate the Rapidase and the
But that was for planting in soil, I'm assuming, right? Because that probably is a disaster for tissue culture, is it?
It would be a disaster, yeah. So I know, but yeah, that was definitely for soil. So yeah.
Yeah. If I may, I actually would like to ask for an advice. I'm new to tissue culture, and we're trying to buy growth chambers for standard tissue culture. Do you have any advice on or considerations? Which ones?
Yeah, we haven't, to be honest, we've inherited all of our growth chambers, but there is one out there that's, and I don't remember the name of it, but I know somebody who was looking for it that's specifically made for plant tissue culture. And what that means is, with plant tissue culture, you can often have problems with condensation depending on where the plates are. And this particular chamber, it might have been by Percival, was specifically made for plant tissue culture to get around that condensation issue.
Okay. Yeah, well, I'll be at this IVV meeting. I'm pretty sure they probably will be there.
You know what you should do, Tim? I mentioned earlier, we have a discussion forum through PlantGENE.
Mhm.
Amy that's a great place for getting information. We have another discussion forum for our plant tissue culture facility group where I posted a question about LED lights and got really good advice. But if you sign up for the discussion forum and ask, you know, people about chambers for plant tissue culture, I'm sure you'll get some good information. Be
Yeah.
easy.
That's a great idea. Thank you.
Yeah.
I'll follow up on that.
Yeah, it's an email. Yeah. Okay, any other questions in the chat? Amy? Oh, speaking of so I was just thinking sorry, Amy. There was one question that came in through email where somebody asked about uneven germination of their tomato seeds. We haven't had to try this, but I have heard that some people will soak them in water overnight, kind of imbibe them for a while. And we've also come across other species where we're working with species that have not been bred for commercial purposes, and that's one of the selection criteria they might have for seeds to be even germination. And we do experience that, but I have heard for tomato that some people soak them overnight to give them, kind of that jumping off start place of germinating. So you can try that. I don't know who sent that question in, but I remember that question.
You read my mind, Joyce, because
Oh,
we were
so
talking
weird.
about germination, and yeah, there's a question that was emailed in from Provere asking about. So the problem is that their tomato seeds germinate at different speeds. So what is the best way to synchronize the germination of the tomato plants? So your recommendation would be an overnight soak in water.
That's what I've heard has worked. I mean, we haven't had to try that really ourselves. But I know it can be an issue with seeds, especially if you're working with maybe a wild species. That can happen sometimes, or some other species where they're like, there hasn't been a whole lot of breeding that's gone into it and selection for lines that do have even germination. So, but that's something that could be tried.
Can you hear me?
Yes.
Sorry, my video not working.
Hi, Margaret.
Hi. Have you tried mature leaves?
Have you tried what? I'm sorry. Margaret, can you repeat that?
Have you tried leaves?
Oh, leaves. Leaves do work, but the reason we don't do leaves, I mean, leaves do work, I haven't done it, but I think there are protocols out there that use leaves. And the only reason we don't do leaves is because we're doing so many different genotypes. And it's such a volume. The cotyledon system works better for us because then we don't have to maintain plants and culture. We certainly don't want to use leaves from the greenhouse because working with other systems where I had to, or in chambers, it was, kind of hit or miss with contamination sometimes and having to clean up the leaves. So, so again, if you look out in the literature, I'm sure there are protocols that do use leaves. But again, we don't because of the number of different genotypes we have to work with. The cotyledon system is just best for us.
Thank you.
You're welcome.
One of the questions that got sent in had to do with blot drying the explants. I just wanted to mention that we really don't blot dry them because then that sounds to me that you are going to be damaging the cotyledons.
And taking off most of the agrobacterium.
Yeah,
because we had so Carrie can explain it's not really blotting, it's more draining. But one time, right, you don't want to damage, but one time we had somebody in the group and we showed them how to do the transformations, but then as people go off and it's sort of they don't remember. And we were wondering why she was getting such a low transformation efficiency compared to others in the group. And when we had somebody watch her, it turned out she was doing a great job blotting everything off to get them really dry. But then through the co-cultivation period, there wasn't enough aggro there to do the infection. So you want to make sure that they're not too wet to avoid overgrowth, but you don't want them so dry that you're going to have like little to no transformation.
Mm-hmm.
So you just want to explain a little bit.
Yeah, so I take all of my explants that are incubating in the magenta box, I pour them all out onto the paper towel.
So is it one layer of paper towel?
Two layers of paper
Oh, this
towels.
long, white
If I'm
paper
using
towel.
25 mils of aggro, it's 2 layers of paper towel, and that should be enough. And like the corners of your paper towels are usually dry at that point. So once in a while, if you have a cotyledon that is like curled up a bit and you've got aggro pooled in the middle, then I will literally just dab the inside of that cotyledon on the corner of the paper towel. But honestly, the majority of your cotyledons should not need that, and you should be able to plate directly onto your media plate without any dabbing with the paper towel.
Hello.
Hello?
I wanted to ask about which method do you prefer to determine the efficiency of guide RNA in CRISPR system as a transit expression?
So, sorry, so you're asking about CRISPR construct?
Yeah, which method do you prefer to determine the efficiency of the guide RNA?
Oh, I see. So, right, so at one point we and others were using protoplasts to determine the efficiency, but then that always didn't translate to when we did stables. So what we do now for the guide RNAs is that we use the online tools, we pick those that are at the top, and then we'll use 2 guide RNAs per gene. So that sometimes if one works and the other one, if one doesn't work, the other one does. And then we've saved ourselves time. And then we also will place our guide RNAs about 50 to 100 base pairs apart to really make sure we're getting good inhibit, kind of knockout of expression. But it's just we design the guide RNAs, do the transformations, and then look at that efficiency. We've been lucky with tomato and with ground cherry. We get a really high efficiency of recovery of edited lines. Of course, there are some genes that we're working with that can affect development, and then we don't recover as many. But I think that what your question is, just like before you do editing, are we determining our efficiency of our guide RNAs? We don't. We just go ahead and do the transformations.
What about rhesogenesis agrobacterium transformation in tomato?
I'm sorry, what?
Rhesogenesis
Oh,
agrobacterium.
rhizogenes. Yes, we did some a number of years ago, but we don't do it routinely. It was just one of our projects. Our collaborator wanted us to do some hairy roots of tomato, and it worked well. We got hairy roots from the tomatoes using the cotyledons.
Can you share with us the protocol of this rhizogenesis agrobacterium
We don't have a protocol. Yeah, we don't have a protocol for it because it was just one experiment. And it was a long time ago, so I don't remember if it was maybe something we just found in the literature. So, yeah, I don't have a protocol to share.
Yeah, okay, thank you.
Sure.
Yeah, thanks for your question, Karam. Yeah, so I think I'd written this in the chat earlier, but just Joyce and Carrie can't comment on the efficiency of other people's protocols, and it's gonna, differ between the protocols, and there's a lot of other factors. I do want to just have a quick plug though, if someone on this call does have a really efficient transformation protocol or like a transient expression, feel free to email me. We do review protocols. The steering committee looks through things for quality control. And we do have a web page on our PlantGENE site. Ultimately, we would like to build a repository of really tried and true protocols. So if you'd like to submit, get in touch.
Thank you.
Okay. Yeah, we have about 5 more minutes. Any other questions people want to ask, or if there's anything else in the chat or something from an email, Amy?
Yes. Yeah, so we got a lot of email questions, which was great to hear from people. And yeah, great to continue this discussion around tomato transformation today. So someone had asked, I don't remember if this was something we had talked about last time or not. What do you seal your plates with? And have you tried different things to seal the plates?
Yeah, good question.
So for the incubation period after the transformation, we do use parafilm, but most of the time we use the micropore tape because we found under the lights on the shelf, if they're wrapped with parafilm, the plates get, there's a lot of moisture in there. And then when you go to do transfers, you're just inviting in possible contamination.
And the other thing, the reason that we also switched to micropore tape from parafilm that we noticed with tomato is we got it seemed to be inhibiting the regeneration to it. We weren't getting really good regeneration, and it was most likely due to a buildup of ethylene that was inhibiting the regeneration. So when we switched to this micropore tape, which is made by 3M, I believe, then we got better, better regeneration. And again, these aren't just some things that we try and say, oh, it worked. We always do side-by-side comparisons with a good sample number before we make any change. And so when I originally came here, people were using parafilm. I was concerned about how, not good things looked. And when we tried that in comparison with this micropore tape, we got much better regeneration. So between the condensation issue and also the regeneration issue, the micropore tape solved that. But one caveat is that, you're gonna have to keep an eye on your medium. I mean, we transfer our material to new medium, freshly prepared medium every 2 weeks. So then we don't have to worry about the medium dehydrating. So we use a micropore tape on everything, except for, as Carrie mentioned, that incubation, co-cultivation period where we use parafilm.
That's very important detail. I've done quick Google search. Is it micropore tape 1530-1 or any specific brand?
I believe that is, we use the 1/2 inch. So I don't know, I think that's the number, but honestly, I'd have to look if that's not.
And is it how many circles around the plate?
Oh, just once.
Just once, okay.
Just once.
Yeah. All those little things can make a difference, can't they? It's just the one thing with tissue culture, and people say, oh, that can make a difference. And I always say that's where you're going to fail with plant tissue culture, because it's just, in my experience, it's just the littlest things that you might not think would make a difference. They can.
Yep. Thank you.
Okay.
Another question was, are magenta boxes necessary for tomato tissue culture?
No, but they make life easier.
Yeah.
That's I always see all kinds of, baby food jars or takeout containers and stuff like that. I guess I haven't personally worked with that kind of stuff, but maybe you guys can speak to, the cost benefit of buying something that's intended for this purpose and maybe makes your life a little easier versus some of the cheaper DIY versions.
Yeah, so baby food jars are great for some applications, but since we're putting I don't know how many, we transfer a lot to it, we'd have to use a lot of baby food jars, and they're a smaller container, and our protocol is based on magenta boxes, and even just changing a culture vessel can make a difference between success and failure. I know there are some people that are using other types of plastic containers that are disposable, but we can't in our hearts contribute any more to the plastic waste stream. So
we go through enough plates and with boxes and waste stream, and using the same boxes for years now. And I mean, that's the great thing. You can just use them over and over and over.
Yeah, and occasionally we might have to replace lids, those get brittle over time.
With the light in the culture room.
So for us, it's, it's not, again, again, there are other people using other containers, but just please be aware that if you do use a different container, you might get different results than what we're getting. So you might have to change some things. And again, it's a preference. And for us, the magenta boxes are a preference for the reasons I mentioned. We can reuse them. We're not discarding them. Because like we said, we already put so much in with this Petri plate, so.
Yeah.
All right, looks like we hit 2:00.
Yes, we are at 2:00. So I will just kind of end the meeting here. First, I just want to say thank you to Joyce and Carrie for joining us today for this follow-up session. We had sent out a survey, after our first discussion, and it was like 92% of people who had responded wanted a follow-up session. So I think that just speaks to kind of the need to have these types of discussions. And yeah, that's I think really at the heart of what PlantGENE is all about, is getting everyone together to have these kind of discussions and hopefully really make a difference in people's research and their outcomes. So again, just thank you to everyone in the audience. Great to see everyone here today for this discussion. And yeah, thanks, Lawrence. Lauren said, hopefully see you guys in St. Louis. So we will all be in St. Louis very shortly for the Society for In Vitro Biology meeting. So yeah, come say hi. Other things that I want to plug. So if you are not signed up for the discussion forum, that is an opt-in thing. It's not automatic when you register to become a PlantGENE member. So if you want to join, there's information on the website. This event has been recorded, like all of our events, and I'll be posting the video online shortly, along with the PowerPoint slides. And yeah, if there's anything else, please get in touch. We have our PlantGENE email, and my personal direct email is also posted on the website. So feel free to drop us a line. And again, just thank you, everyone, and see you at SIVB.
Thank you. Yeah, looking forward to it. Yeah, great.
All right. Thanks, everyone. Bye. PlantGENE would like to thank the National Science Foundation for their support. If you would like to learn more, please visit their website at nsf.gov.